The idea that probiotics could enhance what some researchers now call the neurobiotic sense, the gut's ability to detect microbial and nutrient signals and rapidly transmit them to the brain, marks a radical evolution in how we think about both probiotics and sensory perception. No longer confined to vague promises of digestive health, next-generation probiotics are being designed with a far more ambitious goal: to restore or enhance communication along the gut-brain axis, especially via specialized gut cells called neuropods. These gut-brain intermediaries act much like sensory neurons, detecting microbial byproducts and nutrients, then firing signals directly to the brainstem via fast synaptic connections. The potential to modulate this system with precise strains of beneficial bacteria could unlock powerful new therapies for weight regulation, mood stabilization, and even cognitive resilience. What makes this neurobiotic sense unique is that it operates both below conscious awareness and at remarkable speed, within milliseconds. Unlike traditional hormonal pathways, which take seconds to minutes, neuropod-initiated signals can rapidly alter how we feel, whether we're hungry, and even whether we find food rewarding. These signals rely heavily on inputs from the gut microbiota, which release neuroactive metabolites such as short-chain fatty acids, SCFAs, gamma-aminobutyric acid, GABA, tryptophan derivatives, and bile acids. The right probiotic strains can significantly shift this chemical environment, priming neuropods to transmit signals more accurately or powerfully. One promising candidate is Lactobacillus rhamnosus, which has shown in preclinical studies the ability to influence GABA receptor expression in the brain via the vagus nerve. Another, Bifidobacterium longum, has been associated with reductions in cortisol and improvements in emotional resilience in human trials. These strains don't simply colonize the gut. They alter the information architecture of the gut-brain interface, potentially recalibrating how neuropods interpret the metabolic state of the body and how they communicate with the central nervous system. In the context of weight management, this is especially relevant. Hunger is not merely a matter of empty stomachs or willpower. It's a complex negotiation between nutrient-sensing gut cells, microbial metabolites, and brain circuits tied to reward and homeostasis. Certain probiotics may help restore proper satiety signaling by promoting the release of GLP-1 and PYY, hormones known to reduce appetite and improve insulin sensitivity. Others can enhance vagal tone, increasing the strength and clarity of gut-brain messages that reinforce fullness and satisfaction. When neuropod signaling is strong and accurate, the brain is more likely to receive clear "stop eating" messages, potentially reducing overeating without the need for cognitive restraint or deprivation. Beyond satiety, probiotics may influence how pleasurable or compulsive eating feels by altering the microbial outputs that interact with brain dopamine pathways. This could be crucial for people who experience food as overly stimulating or addictive. If specific strains can reduce the intensity of those reward signals or improve prefrontal regulation over impulsive behaviors, they could become valuable tools for people struggling with obesity, binge eating, or metabolic syndrome. Even more advanced are the emerging synthetic probiotics and engineered microbial consortia that are being designed to perform targeted biochemical functions, such as producing human-like neurotransmitters, enhancing specific nutrient absorption, or delivering anti-inflammatory payloads to the gut lining. These next-gen therapeutics could be tailored to rebuild a precise microbial environment optimized for healthy neuropod activation. Imagine a custom microbial formula that restores lost SCFA production, reduces inflammatory flagellin, upregulates satiety hormones, and boosts vagus nerve firing, all without pharmaceuticals or surgery. Of course, there are caveats. Probiotic effects are often strain-specific and context-dependent. What works in one person may do little in another, especially depending on the state of the existing microbiome, diet, and genetic background. Many commercial probiotics contain generalized strains with low survivability or insufficient potency to influence complex gut-brain networks. And while rodent studies have shown profound effects of certain strains on behavior and metabolism, translating those findings into consistent human outcomes remains a challenge. The neurobiotic sense is not simply a switch to be flipped. It is a dynamic, adaptive communication system influenced by dozens of environmental and biological factors. Still, the evidence is growing that targeted microbial modulation can fine-tune the gut's sensory capacity, recalibrating hunger signals, emotional responses, and behavioral tendencies. If so, probiotics, especially next-generation formulations, could become powerful tools not just for gut health, but for reshaping the way we think, feel, and act in response to food. They may help restore the integrity of a sensory system we didn't even know we had until recently, one that influences our most basic drives and decisions. And in doing so, they may provide a foundation for sustainable health interventions that work with the body's own signaling infrastructure rather than against it.